Well, thank you all for coming. I'm Rush Holt, one of the co-chairs of the caucus. And, this caucus exists for a couple of reasons. The general reason is to promote research in the biomedical sciences. It was connected with the effort in past years to double the funding for the NIH. And it is generally to talk about the importance of this, and the principal way of doing it is by showing the fruits of that research. And today we'll be hearing about a very topical subject. I don't know how many of you see the mail or go around the district with your members, but there are few subjects that rival autism in concern of families today. It is growing. The incidence of autism is growing, and families are scared. It's epidemic, and it's mysterious, and this is the sort of thing that we should be supporting. And so we have an excellent speaker. I won't take your time to talk about her now, but I just wanted to say that from this, you should listen closely and report back to your offices on what we are doing and what remains to be done in understanding, treating, and mitigating autism, and supporting the federal programs that help make that possible. So I'm delighted to see you here, and with that, let me turn you over to, June Goldberg from the American Society of Cell Biology.
Thank you.
Thanks.
Hi, I'm Joan Goldberg, the Executive Director of the American Society for Cell Biology. I wanted to add my welcome. The Congressional Biomedical Research Caucuses have been on the Hill since 1990. Congressman Holt is one of our co-chairs, and we're very grateful for his commitment. We're also very grateful for today's speaker. Dr. Jacqueline Crawley is speaking on behalf of, I would say, NIH researchers, but also, patients. Dr. Crawley received her BA in biology from the University of Pennsylvania and then her PhD in zoology from the University of Maryland. She went on to do postdoctoral research at Yale, and she's currently chief of the Laboratory of Behavioral Neuroscience of the Intramural Research Program. That's the research program that takes place on the NIH campus. She has also been past president of the International Behavior Neuroscience Society. I'm assuming that's past president. And also received one of their highest awards, the Marjorie Myers Lifetime Achievement Award. She's the editor-in-chief of the journal Neuropeptides and has also served on the editorial boards of 10 scientific journals. She's also author of a book called What's Wrong with My Mouse? Behavioral Phenotyping of Transgenic and Knockout Mice. And that sounds like a foreign language to many of you in the room. Dr. Crawley will explain to us why mouse models are so important in understanding autism. And she'll also address what Congressman Holt talked about, the importance of NIH funding to unraveling some of the mysteries about autism, addressing both the extent of the problem and, I think, pointing us towards some areas of hope in the future, which is what science is so important for. So without anything further, I'd like to welcome Dr. Crawley. Sorry, Crawley.
Thank you so much, Joan and Lynn, and I'd like to thank Drs. Bishop and Varmus. It's really an honor to be here, and I appreciate the invitation to come and tell you a little bit about how scientists in laboratory settings are developing strategies to understand the causes of autism and to eventually try to develop effective treatments. So autism is a neurodevelopmental disorder, meaning it starts very early in life. And at this point, the cause remains unknown. We now have very good diagnostic measures to correctly diagnose individuals as either having autism or not. And the main one, ADOS, the Autism Diagnostic Observational Schedule, is what you want to send the children to rather than just a more general psychologist to really be sure that you are, in fact, diagnosing correctly. So the current diagnosis, which was only in place since 1994 in the DSM-4 criteria, has 3 categories of symptoms. The first is deficits or abnormal or unusual reciprocal social interactions.
Yeah, can everybody hear in the back?
Yes. So are we okay?
Yeah, I think it was
Okay.
hard, so
Great.
we may need you to talk to the
Perfect.
mic. Sorry about that.
So the 3 diagnostic symptoms of autism really all have to be met to be considered true autism. So the first is the criteria of unusual or abnormal reciprocal social interactions. This is from a book by Uta Frith in London. The aloof is probably the characteristic that you hear the most about. People who sort of avoid others, don't want to be touched, don't make eye contact, don't have friends, or not involved in any social interactions. The second, the passive, is a little less common. This is people who don't mind being involved in a social situation but just don't initiate anything. And the third, the odd, shows up in a smaller number of cases in which the interactions are extremely awkward and unusual. Often the child will come up and violate your personal space and touch or initiate unusual conversations. And these are the kids that are often considered just strange that you might meet in the real world. So that's the social first diagnostic domain that usually is the main one that's most specific for autism. The second is deficits in communication. Usually this involves early or delayed language problems. And in adults, often the language is very concrete and simple, and all of the nuances and jokes and humor and body language that's part of what you're understanding when somebody talks are not being perceived by the autistic person. So for example, the mother is saying, "Can you pass the salt?" and the child answers, "Yes, I can pass the salt," but that's not the point of the question. The third diagnostic symptom is a group of behaviors, including repetitive behaviors, such as motor stereotypes, hand flapping, and toe walking, repetitive actions. I've watched children who will put together the same jigsaw puzzle over and over again, or draw the same picture many, many times in a row, or want to watch the same scene from a movie, just that scene, that 5 minutes. Insistence on sameness is what a lot of the parents come in with as the biggest problem in the family environment. So if you've seen the iconic movie Rain Man, where Dustin Hoffman portrays an autistic adult, he wants to have the same meals every day and watch the same TV show, and if he doesn't see Judge Wapner at 5 o'clock, he has a temper tantrum, and that's very common. You drive to school by a different route, and the child gets really upset. And so that insistence on sameness means something important that we're trying to understand. And then this narrow, restricted interests, especially for higher functioning autistic individuals, often focus on things like baseball statistics or calendar abilities or a particular video game or collecting something. But in fact, the narrow, restricted interests can be in areas like mathematics or music or medicine or science, and those people are very successful because they're highly focused and that's what they really care about and are good at. And often those lead to special abilities such as mathematics, in which people are said to be savants. They're really very good at those. So there's also, in addition to bona fide diagnostic autism, these other autism spectrum disorders, including Asperger's, in which the language impairment is not so great, although often people are talking in monologues rather than interactively. And then this pervasive developmental disorder not otherwise specified, which is more of a catch-all diagnosis. There's comorbidity, meaning some people with Fragile X mental retardation are also diagnosed with autism. And similarly, these other syndromes, Rett, epilepsy, tuberous sclerosis, Smith-Lemli-Opitz, Timothy, and several other genetic disorders often show some percentage of those individuals often meeting the diagnostic criteria for autism. And again, this is giving us clues about where to look for the brain dysfunctions, what might be overlapping between these problem disorders. Okay, what are the causes of autism? We do not know the answer, but the strongest evidence at this point is genetic. And let me try to walk you through what that means. The ratio of boys to girls is 4:1, which usually means there's some involvement on the X chromosome. The concordance between monozygotic twins is the highest for all major mental illnesses and perhaps for all illnesses, all human diseases that are genetically based. If one identical twin is autistic, the chance that the other will be or have an autism spectrum disorder is as high as 90%. And that is extremely high. And that's as compared to dizygotic fraternal twins or siblings in which the concordance is 5 to 10%. So that differential tells you that it's something in the genome that's similar between the identical twins and different between the siblings. That must be the genes that are involved. And so the general population is even lower. So therefore, there has been a tremendous search funded by NIH research to try to uncover what the genes for autism are. And the two approaches are linkage and association. Linkage studies are like big family pedigrees, and there have been many chromosomal loci that have been identified in various consortia of huge numbers of families with autism and related disorders. These are some of the loci that have been found on different chromosomes. And then the association studies are whole genome scans of individuals. And those have also identified a large number of genes, including a GABA receptor. These are neurotransmitters, chemicals in the brain, developmental genes in the brain that are involved in brain development and connections between neurons. So we have a lot of candidate genes at this point, and researchers are gradually, systematically moving through those one at a time to understand what those genes do and how they might be causing brain dysfunctions that would be producing those 3 diagnostic symptoms. The truth is that none of these genes show up across the board in all individuals. So none of these are clear candidates that show up as a marker for everybody. And in fact, most of them just show up in a few families or a few individuals. So we have a large number of candidates, and certainly this is not a single gene mutation. It's going to be a group of mutations that show up, or individuals have one versus the other, and somehow those all play into some problem in brain development. And so that's where the research field is right now. How do you go about looking into those candidate genes and understanding what their functions are? One very strong strategy is to put that same mutation into the mouse genome and see what the consequences are of the mouse having the same human allele mutation problem. And so mice have been used in biomedical research for many years to look at what the outcome of a genetic mutation is and whether that resembles the human syndrome and what can be done in the mouse model to develop treatments. And so we and several other labs are now engaged in setting up mouse models of autism that are based on introducing those candidate genes into mouse genome. Once you've got the mouse, what do you test for? Okay, so what behaviors would you look at in mice that would have analogies to those 3 diagnostic symptoms in humans? That's the big challenge, and that's what our lab does. We've worked on many neuropsychiatric models, Alzheimer's, depression, anxiety. And so I'm going to show you a few mouse behavioral tasks, what they look like, how they might be relevant to autism, and then some of the data that have been emerging supporting those tasks as potential ways to analyze candidate genes for autism. Step 1 is don't anthropomorphize. We know very well that there will never be anything with this name. We don't even use those words in the lab. We wash our mouths out with soap and water if anybody says autistic mouse. Instead, what we're doing is generating behavioral assays that have analogies to the symptoms of autism. We're never going to capture the actual human symptoms, but we can capture analogies. And the goals of these models then are to test hypotheses about the various potential causes of autism, including genes, other things like environmental toxins, immune function that have been proposed as being involved in the etiology of autism. once we've got a good model, and it actually does have a robust phenotype, robust traits that are analogous to the symptoms of autism, that becomes a translational tool to assay potential treatments, drug treatments, environmental treatments, special education program types of treatments, and to see what will be effective in reversing the consequences of that mutation or that toxin or whatever in our mouse model. So that's the general strategy. So here are the assays that we're working on at this point. And I also brought several reprints from our lab, which I've left right here if anybody wants to take one at the end, that describe some of these tasks. And certainly our website and publications that are available can tell you more about these if you're interested in. So in terms of the reciprocal social interaction deficits, we are looking at social behaviors in mice. Luckily, they're a very social species. They do a lot of interaction, both as juveniles and adults, and I'll show you what some of those look like. Communication impairments are more difficult. Obviously, we can't have a language phenotype, but mice do seem to have a lot of olfactory and vocal cues that I'll show you a little bit about, and respond to each other's cues, and perhaps that's the way mice communicate, and we can tap into those as assays. In terms of the repetitive behaviors and stereotypes, stereotypies, and upset to change in routine, restricted interests, mice actually do things like that. They have perseverative spatial habits that are hard to change, some motor stereotypies, which I'll show you, and restricted exploration of novel environments is easy to measure in mice. So here's the first social task that our lab developed based on the large behavioral neuroscience literature that's already been in place. We modified it to very specifically focus on the types of reciprocal social interaction and social approaches that are deficient in autism. This is a 3-chambered box automated by the NIMH shop. And we basically put in a subject mouse in the middle and give that subject 2 choices. The door is open, and they could either go to this side and explore a novel object, which mice love to explore something wired. This is just an inverted pencil cup with a weight on the top. Or come to the other side and spend time with a stranger mouse, a novel individual that they've never met before, which is usually a big attraction for mice. So here's what it looks like. Our subject usually starts in the middle, immediately comes over, walks around and sniffs around the other individual, and spends much more time on this side with a novel social partner than this side with a novel object. And that's our normal measure of sociability. So a normal mouse, the C57 black 6J, is an inbred strain that's commonly used in behavioral genetics. Will spend more time in the red bars with the stranger mouse than in the green bar with the novel object. And the center is in the white. This happens to be another inbred strain of mouse that we've discovered, BTBR T plus TFJ, that has a lot of autistic-like symptoms, and I'll use those as an example. So here, the time spent with another animal is the same as the time spent with a novel object, or perhaps less. And that's what we would consider low sociability, analogous to low sociability in autism. We also measured the actual interactions. We scored this by hand. This is called nose-to-nose sniffing. And again, the black 6 shows much more sniffing of another animal than a novel object. And this BTBR does not show any difference, statistically significant difference between time spent actually physically interacting in a social way. We then have set up tasks to look at juvenile play, to try to look at earlier stages of development, which is when autism is usually diagnosed. And this is an arena in which we have 2 mice together who are interacting. And again, the black 6 strain does a lot more of these crawling over and under, physically touching each other as compared to the BTBR. This is the work of our postdoc Hewlett McFarlane. A lot more nose-to-nose sniffing, which is probably how mice greet each other and maybe analogous to eye contact or greeting, verbal greeting. And again, this black 6 is high and the BTBR is low. Social grooming is something that mice do. They groom each other's fur. And again, this strain is much higher than this one on social grooming. We also look at a bunch of control measures to make sure we're not dealing with an artifact of the animal being sick or not being able to smell that there's another guy there or see that there's another guy there. And so we make sure that there's no physical or procedural deficits that would be causing a confound in interpreting the social deficit. And these are routinely done in animal modeling labs. The second core symptom, communication, is the one that's really a challenge and what our lab is most excited about actually right now. We have ways to look at olfactory communication. We put a cotton swab down into the mouse's cage and put different odors on it and measure how much time he spent sniffing as a measure of how interested he is in olfactory cues from other animals. So pheromones from the other mice might be on that, and you get a lot of sniffing to pheromones in mice. This is time spent sniffing the same 3 animals, the swab from 3 animals being the same here, and then a new animal here elicits more sniffing, so we show they can tell the difference between animals, and the height of these curves tells us how interested they are in spending time with that olfactory cue. Auditory communications are in the ultrasonic range in mice, and there's very sophisticated microphones that translate ultrasonic to our hearing range and allow us to count how many calls and what are the qualities and properties of the calls of mice. And so here's a pup separated from its nest. It calls at very high, loud rates, and the parents use that call to locate the pup and bring it back to the nest. We found that these black 6 mice show these normal vocalizations when the pups are separated, and these BTBRs actually produce a very unusual call with a lot of harmonics that might be like a kind of guttural call that you hear a lot with young autistic children. I'd like to play for you what mouse calls actually sound like. This is mice in one cage calling to mice in another cage. Oops. And this was a real revelation to us. These are the waveform patterns of the calls. And I'll play one more for you. Sorry to hold you up here. So this is one mouse sniffing the pheromones from another mouse, that same olfactory task I mentioned. And these are the waveform patterns that you're hearing. Does that sound like anything anybody's ever heard before? It sounds like birds chirping to me. And in fact, birds communicate a lot with their calls and teach each other songs. And so this is actually a very new field of mouse communication, and we're now studying what situations the calls are emitted during, and whether there are specific syllables or vocabulary of mice that we can relate to their social behaviors and use as a marker for the kinds of language problems that are seen in autism. The third category, I'll show you a little bit about restricted behaviors. So mice will explore a novel environment. This is a hole board. They stick their noses down into holes, and we can put objects or smells underneath. And most animals will spend a lot of time in all of the holes. And we would predict that a mouse model of autism would have restricted interests and spend most of its time just in one place, which is what you see with children in a classroom. The autistic child is always just focused on one toy train and not exploring the entire room. We also look at position habits, that same concept of wanting to use the same route to school every day. This is a task where mice start here, and there's a food reward on the left. We train them up, and then we switch the food rewards on the right. Can they make that reversal? Or this is a swimming pool for mice in which they have to climb out on a platform to get out of the water. And most mice learn this after a few trials. The platform's over here. And after this animal learns, he'll go right over to that platform. And once they've established that position habit, we then switch the location of the platform to down here, another part of the pool. And this mouse is very good at learning that new location, that reversal. But some of our mouse models of autism don't effectively learn that reversal or take a lot longer. So that, again, would be this concept of perseveration or resistance to change in routine. The last, motor stereotypies, is actually sort of interesting. This BTBR model shows a really striking stereotypy called self-grooming. And so these are two BTBRs who will sit next to each other, not do any grooming of each other, but will do very high levels of this very repetitive self-grooming behavior, BTBR being higher than the black 6 strain. And if you just put one of them alone, you'll see, this is our postdoc Mu Yang found, they'll sit there and self-groom for over a minute, which is very unusual for mice. And that's independent of age, so from very young to very adult ages, the BTBR continue to show this high level of self-grooming, where most strains of mice that drops off by the time they're juveniles. So we think this happens to be a model that seems to satisfy all the 3 diagnostic criteria of autism. I'll just finish up with a couple of the wonderful examples of NIH-funded research that have taken those individual candidate genes and put those mutations into mice and looked to see what the outcomes of those mutations are. So this is in GRAIL2, a neurodevelopmental gene that's shown up in several big autism association studies. And Jim Malonig at Medical College of New Jersey has discovered in GRAIL2 as being a quite common mutation in autism. And Michelle Che and George Wagner at Rutgers have in GRAIL2 knockout mouse in which they've looked at some of these same behaviors. For instance, this is juvenile play being very low in the GRAIL2 mutants as compared to their control group. So we're sort of gradually, systematically going through these candidate genes. Others that have come up in various autism association studies are synaptic genes that are involved in the connection between nerve cell 1 and nerve cell 2 and the communication between 2 neurons in the brain. And so these neurexins and neuroligands and shanks are involved in controlling the neuronal connections as they're developing. And this is a mutation called neuroligand 4 that's shown up in several autism studies. This is a group, Germain in the Pasteur Institute in Paris, and another group, Niels Broes in Max Planck in Germany, showing that these neuroligand 4 mice also spend much less time interacting with each other. This is our social task, in which the normal mice show more time with a stranger than with a novel object, but the neuroligand 3 knockouts show the same amount of time with a social and non-social object. And they also have unusual communication in these guys with those vocalization tasks. So I think we're getting already, with just the very first few studies that are emerging, some actual mouse models of autism that can be used to look for the outcomes of those brain developmental genes when they're mutated. And so I'll just finish up that what we're up to at this stage is developing the right models, developing the assays to study the outcomes of the mutations, to test hypotheses about the potential causes of autism in terms of genes, neuroanatomical problems, immune functions, environmental toxins, and so on. And that once we've got a robust model with a good, strong phenotype, a good, strong trait that we can reliably assay in the laboratory, those then become ideal model systems to try some of the treatments that have been proposed, both behavioral interventions and hopefully eventually therapeutic drugs that will in fact target the initial problem in the brain's development. Thank you very much.
I wanted to open it up for questions and start with you.
All right. Maybe I missed what I think is a central point. When you're talking about the BTBR
Very
mouse
good.
and the other, when there's a difference in sociability and
Yes.
repetitiveness and so forth.
Yes.
What is the genetic difference between those mice? Does it include any of these signatures that you find in one or another
Yes.
humans with autism?
That's a wonderful question, Congressman Holt. In fact, these inbred strains are naturally occurring lines of mice that are homogeneous background. And we in other labs are now engaged in studying the background genes in the BTBR that are responsible for their unusual behaviors. We do not know which ones they are yet. We have 2 or 3 candidates. One is a gene for kynurenic acid, and another is a DISC1 gene that's shown up in some schizophrenia pedigrees. And we'd have a huge grant now being reviewed. We hope we'll have good news to try to really look systematically through the genome of the BTBR and discover what all the unusual genes are.
Now, you went through a list earlier. How many human genetic signatures are there that one researcher or another believes is associated with autism? Is it 1 or 2 or 5 dozen? I don't know, what is it?
So if I count up the number of genes that have come out of the human studies is probably about 30 to 40 right now, depending on how strong the evidence is for each. And the number of mouse models right now, I guess, is in the range of 10 to 20. And there's lots of labs that have applications in right now to develop, grant applications in to develop new mouse models based on the humanized gene. So the idea is you take the exact mutation that's the human sequence and insert that into the mouse genome. It takes about a year to make the mouse, and then maybe another year to do the behavioral testing.
But before you do the insertion, you're going to look at the BTBR mouse
Yes.
and find out how many of those signatures
Yes.
you find.
Those are parallel experiments.
Okay.
Exactly.
Yes.
Thank
Or
you.
someone is going to look. That
Yes.
is. Okay.
Yes.
Okay. Thank you.
Yes.
Have you manipulated their developmental environments to see if they're in a more social environment? Are they all raised in like white boxes, like this lab are, or are they socially interacting?
That's a terrific strategy. So our postdoc Mu Yang is now housing BTBRs at young ages with black 6 mice, which should have normal social interactions, to see if having a sibling that's very interactive will help rescue the phenotype. It's a really good experiment. And it looks promising so far. Yes?
I want to thank you for your excellent presentation. I'm Lee Grossman. I'm with the Autistic Society of America, and we strongly support the efforts of the NIH and what you're doing, and would encourage all of your representatives to get behind the funding for more research into autism. I have a question for you. You mentioned at the beginning that the causes of autism spectrum disorder, that the strongest evidence is genetic. Can you describe or explain how that may relate to the increase that we're seeing in autism?
So I am not a clinical researcher, and I'm certainly not a policy person involved in doing the epidemiology of what's been called an epidemic of autism. But I did bring a slide I can show about the issues here. So this is the kind of curve that Dr. Grossman and others have shown many times, that you have this increasing number of cases of autism in the past few years. In fact, if you look at the reasons behind those curves, the diagnostic criteria for autism were published in 1994, and that's almost exactly when this increase started. So if you didn't know what you were looking for, you didn't have a way to detect a new disease, then of course you're not going to have a diagnosis. So a lot of times autistic children were diagnosed as mentally retarded, and a lot of times autistic adults were diagnosed as schizophrenic. And so once the diagnostic rating scale came out, those diagnoses were changed. There's also much more of awareness, thanks to many of the groups and media publications, that autism is a real separate diagnosis, and pediatricians and teachers are starting to notice it and send their children in for more careful analyses. And so, in fact, my understanding of the literature is that what we're looking at is better diagnosis and better awareness among pediatricians and teachers, and also better special education and services are now available, also in many cases thanks to congressional funding and state funding. And so that these allow parents to actually have some hopes that they can have interventions early on that will help their children, and therefore they're bringing their children into the clinics and to the special services at a higher rate than they might have in the past. So I don't know if you would like to make your own comments on that, but this is where I see the field.
Well, yeah, we do use that chart a lot in the Department of Education. And it seems as at least our data is showing that when you add up the awareness and the increase in population spikes, et cetera, that it still doesn't come close to the increase that we're seeing in autism. I mean, I'm not going to sit here and speculate on what the causes are. There's certainly many theories out there, but I guess I would disagree with the fact that it's just based on increased awareness and expanding of the definition. Those have all played into the fact that there has been a rise, but I think it goes well beyond that.
I would just make a comment. Here, actually, in this caucus, we've had, in past years, I forget how, a few years ago, presentations on this subject. And at least you can say a good case can be made that it is not just diagnostic. I think there are strong counter-arguments, but it really is confusing.
I was wondering about the comment on Lee's question. I think that many people in the field, in the autism research field, are coming to the sort of consensus that a large part is genetics, but there's also the environmental interactions. So in fact, not just in the field of autism, but cancer as well. You know, people are prone to certain kinds of cancers, but there are environmental risk factors that will sort of promote the cancers in one individual or another if they have the genetic risk genes that you know, subject them to this higher risk. So I think it's a gene by environment interaction that might help to explain the possibly increasing incidence of autism. The problem is we don't know what the environmental factors are. I think that helps to kind of reconcile what you said.
So Dr. Hu is right, yeah, and we were both just at the International Meeting for Autism Research in London last month, in which there's a presentation from California researchers on some of the agricultural areas, San Fernando Valley and Salinas, in which there's a very high incidence of autism, and so they're looking into whether there are environmental pesticides being used in farming that might be contributing factors interacting with genetic factors. So these are all very important hypotheses to be tested at this point. And the autism field is sort of like where cancer research was 50 years ago, or where Alzheimer's research was 20 years ago, in which we do not understand the biology at all. And there are all kinds of good ideas out there, and just they need to be one by one either refuted or supported by the research evidence.
Doctor, you mentioned in your presentation that you and several other labs have grant applications into the NIH. How has the slowdown in NIH funding over the last several years impacted?
It's really bad. This is a really bad moment in NIH funding, as you all may know. And, you have to get in the top 10%, top 10th percentile or better. It's usually the 7th percentile to really get funded. I'm in the intramural program. We don't compete with the university people for NIH grants, but I'm listed as an unfunded collaborator in many cases, and so I know what their success rate is. They're fabulous grants that are not getting funded. So.
In this particular area?
Oh, yeah. And luckily, some of the foundations, Autism Speaks and others have, Simon's Foundation have raised a lot of private money that is being used for research at this point, and they're funding a lot of smaller grants, which is helpful. Gets people over that hump until their NIH grant can get funded. Yes.
I have a question. I'm representing Autism Speaks, but I don't have a specific question about the issue of incidence or prevalence. So if you actually have CDC, they actually have not disaggregated the diagnostic data to determine what the baseline is. So back to what Congressman Holt said, I don't think the diagnostic issue has been determinative of the increase, and there are other factors that increase it. My question relates to, as a scientist, this vaccine compensation fund case regarding Poland, which, if I recollect, I'm not a scientist, they determined that the child had an acute mitochondrial defect, that autistic-like symptoms were triggered by an excessive administration of vaccines. Now, I'm not into the Marisol issue at all, but couldn't, are mitochondrial defects, are they inherited, or they can occur spontaneously? And are there particular segments of the population who are predisposed to getting autism who may have this mitochondrial defect?
Yeah. This is completely outside of my area of expertise, the biology at least. The case that you're describing, my understanding is that this young girl had about 5 different vaccinations within a week period. So she mounted a huge immune response, very unusual. And that's different than the people who are talking about the single vaccine at age 2. But there's a huge amount of interest in immune function involvement in autism, prenatal infections as well as vaccines. And again, I think this is a very early stage of doing the medical research and finding out A, what the facts are, and B, how immunology could influence autism. And your question about mitochondrial genes is a wonderful one that, totally outside of my field. But it's certainly, another experiment that needs to be done.
Do we know how prevalent that mitochondrial defect might be in the population?
Maybe somebody else here does. I sure don't. It's a good question.
Jackie, those vocalizations you played, they're very interesting. Those are from the black 6 mouse, the wild type, the more social mouse?
The one I just
Do
played.
you have, I'd love to hear the BTBR.
Yes.
Do you have a little snippet
Let's
of
do that afterwards, yeah.
Oh, okay. I want to, see how distressed they sound compared to the other ones.
So our postdoc, Maria Luisa Scattoni, has just finished a very big study and characterized the calls of mice into 10 different categories. Rising, circumflex, long, short, and we're trying to work out whether this is actually the vocabulary of mice. And these are individual syllables that will show up across strains. And she just has a paper submitted in which the four strains that we looked at, 3 show all 10 categories, and the BTBR only shows 6 categories, which may mean it has sort of a restricted language if it turns out that that really is how mice are communicating.
But it's all over the sun.
Yes.
We're not hearing this in our attics.
No. Mice, yes, correct. Mice do squeak if they're stressed, if you pick them up, if they have some painful or predator response. You can hear squeak sometimes, but most of it you can't hear at all. Maybe your dog can. Or your cat. Yes.
It looked like you had a slide on vaccines and autism, and so if you have more to say about that.
So this is a very controversial topic, and I want to tread very carefully. And I brought a slide from the IMFAR meeting that is commonly seen. So this is, again, time, and this is, again, number of cases of autism. And as many of you know, the issue was thimerosal, a preservative in some of the vaccines, contains mercury as well as antibiotic. And there was a very real possibility that that was involved in the cause of autism because a lot of people get their children vaccinated about age 2, and the diagnosis of autism often occurs around age 2 or 3. So there's a temporal coincidence between when the vaccine happens and when the autism symptoms begin. And so there's a lot of interest in whether those vaccines could be the trigger. And you can see that the incidence of autism continues to rise, whereas the vaccines have had the thimerosal and mercury removed for a long time now. So in Europe since the early 90s, in the US since the mid-90s, those preservatives are no longer in vaccines. And so there's really a disconnect between the rise in cases and the availability of vaccines that no longer have thimerosal in them. So there have been several very large studies funded by NIH and by the Centers for Disease Control, epidemiological studies that show there's no actual correlation between what's in the vaccine and whether a child gets autism. And as we were just discussing, there may be immune responses to vaccines in general that could be issues, but it doesn't seem to be this particular preservative that you might have heard about a lot in the popular literature.
If I may, to talk to that point, I'm with the Coalition for Life Sciences, and together we work with the caucus to bring these presentations. And next Wednesday, we're having the caucus in this room with Dr. Paul Offit, and he will be discussing the safety of childhood vaccines. So if you have further questions, he might be more of the expert to ask, not to take away any credibility.
Perfect. Thank you. I appreciate that.
Sure.
With children, there's you mentioned a period of 18 months, 2 years, where there is a normally behaved child demonstrates autistic symptoms. Do you find a similar lag with your mice in terms of, are they normal to a certain period, whatever mouse years are, and then all of a sudden they change?
So that's a great question and a great experiment. And we're looking at it right now very carefully, trying to do longitudinal studies in mice. And I don't have any answers for you yet. It's just too early in the vocalization project, and the social task, we're just starting to look at it across ages. That certainly would be ideal. And it's important to understand that no animal model is really going to replicate all the components of a human disease. So I don't know whether we'll see like a language regression, which is happening often at ages 1 to 2 in autism. I don't know whether we'll see all of the symptoms in all of our models for each of the genes. And so, we try to be cautious. We understand what a model can and can't do. We don't often see the 4-to-1 male-female ratio in our mouse models. Often that BTBR, we see about the same amount of traits in the females as in the males. So, we have limitations in models, and I'm not sure we're going to get to exactly where you're hoping we will, but even if we can just get a partial symptom appearing in the mouse model, I think that then becomes a useful assay for treatment efficacy.
One of autism's core sort of conditions, I guess, is the difficulty in communication and sort of taking the literalness of what people say.
That's right.
I don't know if that's the right word. And you can't really attribute what people actually mean in their intention. Like you gave the example of passing the salt. And based upon my understanding is that mice can't really do this regardless of their symptoms. Like only about 5% of their brain is actually dedicated to that, if less. So how do you really account for this in your studies?
Yeah, that's a wonderful question. So I think what you're getting at is the concept of empathy or theory of mind, that you sort of know what other people mean when they're telling you something, even though they might just use a few words. You get the whole picture. And you can often predict what other people are going to do by their earlier actions. Those are big problems for autistic people. They don't get what other people are thinking or feeling very well. And whether mice ever understand what the other guy is doing, whether they learn from each other, whether they can predict each other's behavior, whether the other guy's behavior influences theirs, those are great experimental questions. And our lab and several others are starting to look at that. So Jeff Mogul at McGill University in Canada has a model of empathy in mice that when one gets a painful stimulus, the others from its cage show a real reaction, whereas others from a different cage don't. So there are going to be experimental ways to address your question, and the field is just gearing up. And I think the autism research community is really driving our motivation for trying to develop these excellent tasks for mice that might be useful.
Well, I wanted to thank Dr. Crawley and also make a few remarks. Lynn referred to the Coalition for the Life Sciences. That's an organization made up of the American Society for Cell Biology, American Society for Clinical Investigation, Genetic Society, Howard Hughes Medical Institute, Society for Neuroscience, and Society for Science and the Public. And so we all work with the caucus to put on these particular caucuses. If you work for a congressperson or a senator and you're not a member and would like to join, and to be sure that you're getting invitations, you can contact the offices of one of the co-chairs. So besides Rush Holt's office, that's Mike Castles, Lois Capps, or Brian Bilbrays. Also, to make sure that you're on our list for future caucuses, like the one on Wednesday next week on childhood vaccines, be sure to contact Lynn Marquis, going to our website, www.coalitionforlifesciences.org. And I also just wanted to thank everybody for coming. Thank you to Dr. Crawley especially. And I think that many people today spoke about the importance of NIH funding, in particular for intramural researchers, for extramural researchers, those who are off campus. NIH funds more biomedical research than anybody else. It's where most of the money comes from, and it's where the cures of tomorrow will come from too. So thanks for your support. Thanks for coming.